Brittany Hood, and this is my project on epithelial cell migration. This project is important because epithelial cells line our body and organs, creating a line of defense. This is crucial in understanding how to maintain our health in the medical field. Epithelial cells are typically found in sheets or clustered, held together by E-cadherin proteins. When epithelial cells leave the cluster or sheet to migrate, a process called epithelial to mesenchymal transition is initiated. During this process, E-cadherin between adjacent cells break their bond and are downregulated, relocalizing in the cytoplasm to be degraded in lysosomes. This then allows the cell to break off and migrate. Pressure is known to regulate this process, but is not yet clear how it affects E-cadherin relocalization. Based on this information, the following question is asked. Do low-pressure conditions relocalize E-cadherin within the cell, thus initiating EMT? It was hypothesized that not only does intracellular pressure control E-cadherin relocalization, but low internal pressure would have the greatest effect since low pressure is associated with increased migration. Below you can see a flowchart describing the methods used this summer. Identifying migration patterns and structures is displayed under microscopic visualization of subcellular organization. In this image, the fibroblast is displaying lamellipodial migration. This migration relies on actin polymerization, which is cytoskeletal actin polymerizing into filaments that push against the membrane, creating a protrusion. Actin in this picture is stained in red, the nucleus blue, and cortactin in green. Cortactin is a protein that helps actin polymerize at a higher rate. Areas of yellow show the overlap of actin and cortactin, indicating the front or leading edge of the cell. The next section, epithelial cell analysis, was the study between E-cadherin and internal pressure. Here you can see the sheets of epithelial cells tested in the conditions. Control cells maintained their pressure. Hyperosmotic cells were placed in a media where water left the cell, decreasing pressure, and hypoosmotic cells were placed in a media where water entered the cell, increasing internal pressure. During this analysis, I selected cells and measured the intensity of E-cadherin in the plasma membrane and cytoplasm. The measurements were averaged and ratioed, comparing the plasma membrane to cytoplasmic intensity. The localization ratios are shown in the graph below. A ratio equal to 1 indicates an equal distribution of E-cadherin between the plasma membrane and the cytoplasm. A ratio greater than 1 indicates more E-cadherin in the plasma membrane, and a ratio less than 1 indicates more E-cadherin in the cytoplasm. This graph suggests that there is no statistically significant difference between control and hyperosmotic cells. However, the comparison between both control and hypoosmotic cells, as well as hypo and hyperosmotic cells, is significantly different. Based on this data, it can be concluded that E-cadherin does not relocalize due to decreased osmotic pressure. However, we do see that high osmotic pressure increases the amount of E-cadherin in the membrane. Thus, the initial hypothesis is supported that pressure affects relocalization, but the overall prediction is refuted because decreased pressure does not have the anticipated effect. These differences may have also been affected by the different morphology resulted by the treatments. This could be due to dilution or higher concentration of E-cadherin resulting from the osmotic flow. Some future work includes plectin, which is a cytolinking protein involved in lobopodial migration, also known as the nuclear piston. During preliminary analysis, we explored plectin's role in maintaining nuclear integrity in both 2D and 3D environments. As you can see in the graph below, there is only a significant difference between the two groups of 2D compared to the two groups of 3D. 3D displayed more folding or compromise of the nuclear integrity than as shown in 2D. The picture labeled 2D control displays a round nucleus that was maintained, and the 3D plectin knockdown image shows a folded nucleus that was compromised. Further research will improve the data and allow for the support or review of the preliminary findings. To view the full resources used this summer, be sure to scan the QR code below. I would like to thank Dr. Petrie and his lab team for their incredible guidance and support this summer. I would also like to thank Pragati Chengapa and Donna McKeon for their amazing training and cellular images used for analysis. Thank you to the STAR advisors and community for this wonderful opportunity, and thank you for listening. Please feel free to leave any questions or comments below. Thank you so much.